Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, the question and answer portion will begin, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Hilary Sherman from Corning Life Sciences.
Hello, and thank you for joining us today for this presentation on human mesenchymal expansion, cryopreservation, and DMSO reduction. I'll start today by giving a little background on MSCs and their use in cell therapy. After that, we'll talk about MSC scale-up and some work I recently completed using Corning hyperstack vessels for scaling up bone marrow-derived MSCs. After that, we'll talk about cryopreserving large quantities of MSCs and how the Corning X-Wash system can be used to reduce DMSO concentrations after cell thawing. So since the 1990s, MSC use in cell therapy applications has been consistently increasing year over year. This figure shows registered clinical trials from the first occurring in 1995 to when the review was conducted in 2020. As of June of 2020, there have been 1,138 registered clinical trials using MSCs to investigate therapeutic potential worldwide. These include both autologous therapies meant to treat the patient with their own cells and allogenic therapies which use cells from universal donors. MSCs can be derived from a multitude of tissues, and the source can have an impact on the differentiation and proliferation capacity of the MSCs. The most common source of MSCs used today in clinical trials are derived from bone marrow. Bone marrow-derived MSCs were the first identified and have been consistently relied upon due to their ease of access and robust performance in vitro. The second most commonly used source for MSCs currently would be umbilical cord-derived MSCs. These MSCs are desirable due to their ease of access and promise of faster self-renewal and expansive differentiation capability. In terms of therapies being investigated, there's quite the range. MSCs have demonstrated the ability to reduce inflammation, contribute to cartilage and subchondral bone repair, improve cardiac function, and they show promise in numerous other conditions. One of the main challenges with using MSCs in cell therapy is the ability to scale up enough MSCs. Studies with MSC doses range from under 100 million to 1,200 million cells per patient for a single dose. A typically used estimate of effective MSC dose is somewhere around 2 million MSCs per kilogram, which works out to about 160 million MSCs for a 170-pound individual. I wanted to put that number into perspective, so I put together this slide showing how many vessels would be required in order to achieve 160 million MSCs. In my experience, MSC densities vary significantly based on the MSC source, the media used, and the application. Typical ranges vary from 20,000 cells per centimeter squared to 100,000 cells per centimeter squared upon harvest. This slide assumes a density of 40,000 cells per centimeter squared at harvest, which roughly equates to 18 T225 flasks, 5 roller bottles, 6 layers of a cell stack, 2 hyper flasks, or a half of a Corning hyperstack 12-layer vessel. Now that we've covered a bit of background, I want to share with you some data from the MSC scale-up work that we did with bone marrow-derived MSCs, which we grew in Corning hyperstack vessels. For those of you unfamiliar with Corning hyperstack cell culture vessels, I just wanted to provide a little bit of background. Hyperstacks are specialized cell culture scale-up vessels that are closed system and utilize gas permeable layers for gas exchange to occur directly underneath the cell culture. Because the gas exchange occurs directly through the film in which the cells are cultured, there's no need for empty headspace above the cell culture medium, and that allows for much more compact cell culture vessels. For example, a 12-layer hyperstack vessel takes up approximately the same footprint as a 2-layer cell stack vessel. Hyperstacks are offered in 12-layer and 36-layer options. For our work, we use the 36-layer format, which is approximately 18,000 centimeters squared. Bone marrow-derived MSCs were scaled up using Rooster Bio's Rooster Nourish MSC xeno-free media. Cells were seeded into the 36-layer hyperstacks at a density of 3,000 cells per centimeter squared. After 5 days of culture, the cells were harvested and counted in order to assess viable cell density as well as the cell viability. This was repeated 3 independent times. And you can see from the data that we achieved viable densities between 40 and 50,000 cells per centimeter squared, as you can see by the bar graphs. And the viability was maintained above 90% at all times, which you can see with the dots above the bars. Additionally, we wanted to ensure that the MSCs maintained their differentiation capacity. And so here we're showing representative MSC marker expression from one study. The International Society for Cellular Gene Therapy, or ISCT, has defined the minimum criteria for HMSC quality as expressing greater than 95% of CD105, CD73, and CD90. Additionally, there should be a lack of expression of typical hemopoietic markers, CD45, CD34, CD14, CD79A, or CD19, as well as HLA-DR surface molecules. Here we're showing the sample in blue compared to the isotype controls in black. And you can see that we have very high expression of markers confirming differentiation potential and really low expression of a cocktail of markers that show actual differentiation. Okay, so now that we've demonstrated the ability to scale up quality bone marrow-derived MSC, the next step in the cell therapy workflow is to cryopreserve the MSCs. In order to simplify cell therapy workflows, cryopreservation of MSCs has become a really important step. Freezing allows for an increase in the potential range of administration, an increase in the shelf life, and time for any quality testing that needs to occur. Cryopreservation bags are designed for single-use storage, preservation, and aseptic transfer of large volumes of cells. Cryopreservation bags are made from a unique bag film container that remains flexible at ultra-low temperatures due to being made from a proprietary film blend. Corning bags are offered in 4 sizes, covering a fill volume range of 10 to 190 milliliters. And they offer both membrane ports and velour connections, depending on what the customer's needs are. For our studies, we froze our MSCs in a mixture of 90% FBS and 10% DMSO. The DMSO prevents ice crystal formation, which can damage the cells as they freeze. Unfortunately, the DMSO itself can also be cytotoxic for cells and may need to be removed prior to administration to a patient. And so the process of removing DMSO can be problematic, as many methods are not closed system or can impact the viability of the cell product. The Corning X-Wash system is designed to aseptically remove DMSO from thawed material in a semi-automated closed system. The X-Wash system is a combination of a single-use disposable cartridge along with a control module that contains infrared sensors to detect the interface between cells and media, and thus coordinate cell transfer to a harvesting chamber, which is separate from the main chamber where cells and fluid are initially added. The control module also tracks the entire process to support GMP processing, monitoring, and reporting of information. Using the X-Wash system, volumes of up to 240 milliliters of product can be washed, resuspended, and volume reduced to volumes as low as 3 milliliters. So here's how the process actually works. Cells and buffer are added to a single-use cartridge via spike ports or tube welding. The cartridge and the control module are then added to a centrifuge in order to actually pellet the cells. Buffer is then removed and can be replaced with either another 240 milliliters of buffer if required. Cells are then resuspended in order to actually wash them, and the cartridge and control module are then placed back into the centrifuge where another pelleting step and buffer exchange can be performed. Or cells can be pelleted for transfer to the harvest chamber. Once cells are in the harvest chamber, they can be removed by tube welding or a simple syringe. After washing, we wanted to determine cell recovery and viability of our thawed HMSC. Here you can see that our data shows recovery of greater than 70% was maintained after thawing, and MSC viability remained high. Here, recovery is represented by the bars, and the viability is represented by the dots above the bars. Most importantly, we wanted to quantify the DMSO reduction after washing with the Corning X-Wash. We collected samples from cryopreserved cells as well as final harvested cells and had the samples analyzed by UPLC. Our data shows that we were able to reduce the final DMSO concentration by more than 40-fold. Additionally, more washes could be performed to reduce the DMSO concentration even further. Lastly, we wanted to ensure that HMSCs that went through the X-Wash after cryopreservation, after thaw, after washing, could then be replated and maintain their appropriate marker expression. So we went ahead and replated the cells for several days and then harvested them and analyzed them to ensure that the cells did recover and maintain their multipotency. Here you're looking at average marker expression from 3 independent studies, and you can see that we had high expression of appropriate differentiation potential markers and very low expression of markers that would show differentiation occurring. So in summary, Corning hyperstack vessels offer a nice tool for scale-up of large quantities of MSCs for cell therapy applications. Once cells are scaled up, cryopreserving them in containers such as cryopreservation bags can really streamline the cell therapy workflow. And then finally, the Corning X-Wash system can reduce unwanted cryoprotectants such as DMSO from the cells without negatively impacting MSC recovery or quality.
Okay, so I'm going to go ahead and ask Hilary the first question. Hilary, are you ready?
I am.
Okay, great. So can the X-Wash be used for other cell types?
Most definitely. The X-Wash or X-Series products were actually originally developed for working with whole blood, and so the X-Wash has been used extensively for washing blood-based products such as PBMCs, and as well as other cell types.
Okay. And is there any way to visualize cells on the hyperstack?
Yes. The HyperStack 12 actually can fit under microscope, in which case the light source can swing away. So there's sort of a plastic top plate that's banded onto the hyperstack for easier handling and for holding some of the tubing, but that can easily be removed, and that way the hyperstack can be placed under a microscope in order to visualize the cells. Additionally, if that's not an option for customers, or if they're using the Hyperstack 36, which wouldn't fit under a microscope, there are some handheld based microscopes that can be utilized, as well as using sort of a surrogate vessel, such as the Hyper Flask vessel, seated at the same density, growing under the same conditions, can be used to kind of get an idea of the confluence of the cells.
Okay. And then how much volume could the XWash process?
Yeah, so the max volume that can be placed into the XWash at a given time is 240 milliliters. And so then that can be pelleted, aspirated, and then replaced with an additional 240 milliliters of buffer. And we recommend it's used for up to 2 separate buffer exchanges after that, as the max number of times the vessel can be centrifuged and processed before an additional vessel would be required.
Okay. So it looks like we have time for just one more question, but as a reminder, if you could just go ahead and type in your questions, we can pass those on to Hilary and she will follow up with you directly. If your question wasn't answered, we'll pass those questions on to her. The last question for the webcast is, how much volume of harvesting reagent do you use for Hyperstack?
Yeah, good question. So we recommend about 200 milliliters of harvest volume per module. So we consider a module like the 12-layer vessel. 36 is 3 modules put together, so 36 layers. So I would use about 600 milliliters of harvesting reagent for the Hyperstack 36.
Okay, great. Thanks, Hilary. And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing us a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Bye-bye.